Just like in mitosis, before meiosis 1, interphase occurs. During interphase, there are three separate phases: growth 1, synthesis, and growth 2. Synthesis is the most important part of interphase because DNA replication takes place. When this happens, there are DNA duplicates known as sister chromatids, which are joined together by a centriole.
On to prophase 1, the chromosomes become visible as they shorten, coil, and thicken. That is, as they condense. Also, the spindle fibers begin to extend outwards from the two centromeres, which move to the opposite ends, poles of the cells. So the sister chromatids attach themselves to either nuclear membrane. Each chromosome is composed of two sister chromatids containing identical genetic information. During most of meiosis through the end of metaphase 2, the sister chromatids remain attached to each other at the centromere. During prophase 1, the two membranes of each homologous pair become intimately associated along their entire length as they synapse to form a structure known as a tetrad. The two chromosomes of each homologous pair adhere to each other during this phase of meiosis. The two chromosomes making up homologous pair, however, are not genetically identical because these chromosomes are inherited from different parents. When the chromosomes synapse during prophase 1, which is to say when they combine to form a tetrad, genetic variation takes place in a process known as crossing over. During this process, the two chromosomes of each homologous pair exchange segments of DNA, which, as we can see in this video, will look something like this. As a result, the gene combinations as a chromosome can be changed, and the child that is eventually born ends up with a mixture of both parents' genes in its chromosomes, which is slightly different than its individual parents' genes.
Metaphase 1. In this phase, the homologous pairs which were in the form of tetrads now separate and start to line up along the equatorial plate, opposite to one another. This is where we can expect to see an important part of the cycle, as independent assortment occurs here, and this again creates genetic variation. When the homologous pairs line up at the equatorial plate, they're not required to line up in a certain manner. So it doesn't have to be that all the maternal chromosomes go on one side of the plate and all the paternal go on the other side of the plate. They can line up on either side of the metaphase plate, and this creates variation in genetic information because now the genes that will go to each haploid upon cell division are going to be different. It is possible that a cell may end up with all the maternal derived chromosomes, all the paternal derived chromosomes, or with a mixture of paternal and maternal chromosomes. From there, the sister chromatids are attached to the spindle fibers, and this is where we head into anaphase 1. Now, in this brief phase, the homologous pairs are pulled to the opposite sides of the poles, when the maternal would go to one side and the paternal chromosomes would go to the other. The separation ends up giving each of the cell 23 chromosomes, which is the haploid number, but still with a pair of chromatids. So this is known as reductive division, as the chromosome number will now be half, since once the cell divides, it will go from 46 to 23.
During telophase 1, the spindle fibers disappear. Nuclear membranes begin to form around the chromosomes at each end of the cell. The chromosomes in the two nuclei are not identical, since each daughter cell receives only one member of each homologous pair. These daughter cells are now haploids, which means they contain only n number of chromosomes. Cytokinesis occurs, which is the pinching of the cell membrane in order to completely divide the two daughter cells. This, gentlemen and ladies, is a close-up of one of those cells.
Meiosis 2, we find ourselves in the interphase 2. So interphase 2 is very much similar to interphase 1, but the only difference is that there is no DNA replication as the DNA was already copied in interphase 1. Now moving on to prophase 2. As in prophase 1, the chromosomes condense spindle fibers form and the centrioles begin to separate and the nuclear membrane fragments disappear. But unlike prophase 1, there is no attachment of the chromosomes with the nuclear membrane to exchange genetic information. There are haploid daughter cells, that mean the cells have a single set of unpaired chromosomes as compared to prophase 1 that was diploid and had homologous pairs. Prophase 2 is also a lot shorter than prophase 1.
Now moving on to metaphase 2, this is very similar to the metaphase in mitosis, and to briefly go over it, the chromosomes line up at the equatorial plate and are then attached to the spindle fibers coming from the centrioles. The difference between this and metaphase 1 is that there are no longer homologous pairs lined up at opposite sides of the plate, but just single sister chromatids that are ready to be pulled apart. So of course nothing like independent assortment occurs in this phase either. Moving on to anaphase 2, this is very similar to the anaphase from mitosis. The 23 sister chromatids that lined up on the equatorial plate are now separated by spindle fibers as they are pulled apart and towards opposite poles. This continues to deem the cell a haploid, but now only with one chromatid on each chromosome.
We now enter telophase 2, which is the final phase of meiosis 2. The second nuclear division has been completed, and the second division of cytoplasm follows. The chromosomes at each pole lengthen and decondense. We now enter cytokinesis 2. The cell membrane on each cell invaginates at the equator and creates 4 haploid daughter cells or gametes. To conclude, once their gametes have been formed, one sperm cell and an egg cell eventually combine to form a diploid, known as a zygote. This grows and develops into a human body, which again shows us why this cycle is so important in our lives.